Today's topic is glycogen metabolism. Any idea about glycogen? What is glycogen? Quickly. Glycogen is a storage form of glucose in animals. So the skeletal muscle and liver are the major organs which stores glycogen. Liver stores about 10% of its weight in the form of glycogen, whereas muscles can store up to 2% of its weight by weight as in the form of glycogen. Now, what is the weight of liver? On average, 1.1 to 1.4 per kilo, that's all. Say for example 1.2 kilo. In a 70 kilo person, around 40 kilo is muscle. Muscle is the highest weight. So, which one contains more amount of glycogen, muscle or liver? Muscle. But muscle glycogen cannot be utilized for survival. When you are starving, muscle glycogen cannot be utilized. Muscle glycogen can only be utilized for muscle exercise. Alright? Nothing else. Whereas liver glycogen is very benevolent, dedicated guy. Whenever our body is starving, it will come out and it will supply glucose to the required tissues. Fine. That's why muscle glycogen is called selfish glycogen. What is the major difference between these two? Glycogen, muscle glycogen can contribute to blood glucose level between two meals. Morning around 8 o'clock you had your breakfast. Afternoon around 1 o'clock you will have your lunch. How many hours you have been starving? 5 hours. How long the sugar can last? Almost that much time from glycogen. Now if you skip your lunch and take it to the up to dinner, then it becomes almost 13 hours or 12 hours. So that time when you skip lunch, when you don't skip lunch, your blood glucose is maintained by glycogen. When you skip lunch after post-lunch session, your blood glucose is maintained by gluconeogenesis. Clear? Okay. Timing around 4 to 6 hours, the stored glycogen can last. What about the size of the body, weight of the individual? Does it matter? If the individual is like this fellow, shut up. Just imagine me and her liver. What is the difference in liver? My liver might be half of his body, half of her body weight alone. Right? Almost like that. So, if I can survive 6 hours, can she survive 6 hours with her storage glycogen? Yes or no? Yes. Because I may not survive, because my requirement is more. See, my size of the body has increased, right? Not only vertically, but also, if you are increasing size in vertically, your liver size will increase. The moment you increase horizontally, your liver size remains same, and it has to feed more mouths. So I will feel more hungry than she would. Sit down. But while eating, she will eat more than me. Correct? I will eat more frequently, but she will eat more in amount. That is why she is thin. Her metabolism is much faster than mine. That is why she is thin. She is utilizing more of energy, and I am like a banker, always storing. Who lives a life? The one who spends or one who stores? The storage of glycogen can be seen by staining the hepatocyte like these under the microscope. These are the glycogen molecules, these spots. Okay, they take a light red color spot. The glycogen has got two types of bond, alpha 1,4 glycogen bond mainly and alpha 1,6 at the branch points. What is the difference between amylopectin and glycogen? Glycogen is more branched than amylopectin. what could be the highest frequency of branching in human glycogen? At every 8 glucose molecule. At every 8 glucose molecule. তার মানে কি হবে? হাইয়েস্ট মানে 7 বা 6 হবে না, it can be 6 or 7, it can be 9, it can be 10, but it cannot be below 8. The highest frequency will be around 8. Now, glycogen catabolism, let us break it first, then we will make it. Glycogen catabolism or glycogenolysis. Glycogen catabolism or glycogenolysis is a process or metabolic pathway or catabolic pathway which breaks down glycogen to produce glucose. Correct? Now, this pathway begins with main glycogen molecule. Now, if you can recall the structure of glycogen molecule, this is a field of vision under microscope. So, glycogen molecules will look like this. Correct? Like this. So, it will have one reducing end and a number of non-reducing end. That is why the effective osmotic pressure exerted by a glycogen molecule containing more than 100 glucose is equivalent to exactly the osmotic pressure contributed by one molecule of glucose. Because glucose has one reducing end, it has only one reducing end. Non-reducing end does not contribute to osmotic pressure. So, whatever number of non-reducing end may there be, it is not going to be counted as osmotic pressure contributor. Fine. And that is why it is stored in the liver in the form of glycogen, not in the form of glucose. If it otherwise had it been glucose stored in the glycogen in the liver in pure form, not in glycogen form, then what would happen? Soon after taking food, our liver would have swollen like this, and as the time will pass on, liver would have come down. That never happens. Why? Because of this. So, every structure has got its own function. This is the one structure function relationship. The second structure function relationship of glycogen is, see, we have 5 fingers. Have you ever seen cutting people beans? বড় বটি কাটা দেখেছো কখন? বড় বটি কিভাবে কাটে? Beans. Cutting beans. How they cut? First they make it half and then they place it together in a bunch and then cut together. Why? To save time. Same effect. Similarly, if you have more number of non-reducing end and if you chop it, you get 5 pieces of fingers. If you have only one, then you get only 1 piece of fingers. So, if you have more non-reducing end, then every time the glycogen breakdown will take place, from each non-reducing end, one molecule of glucose will be released. So, when needed, it will produce plenty of glucose in no time. Clear? So, these two are structure function relationship of human glycogen. Now, this phosphorylase enzyme, which is oftenly called as a misnomer glycogen phosphorylase. The name of the enzyme is phosphorylase, only phosphorylase. But many books or many people, many teachers might mention it as glycogen phosphorylase, just to make you understand that it's a part of glycogen metabolism. Okay. So, at your level, we will definitely consider that mistake. So, phosphorylase enzyme require inorganic phosphate, and every time it will release one glucose 1-phosphate and rest of the glycogen chain as it is. See, it begins with glycogen with N residues plus inorganic phosphate forms glucose plus glucose 1-phosphate and glycogen N-1. Now, what happens with this glucose 1? It is converted to glucose 6-phosphate. What could be the enzyme? PGM. नाम क्या है? Phosphoglucomutase. গ্লাইকোজে কি ছিল? আইসোমারেজ, ফসফোহেক্সোজ আইসোমারেজ। ওখানে একটা মিউটেজ এনজাইম ছিল, কোথায় ছিল? 3 PGA to 2 PGA, ফসফোগ্লিসারেট মিউটেজ, একই বানান, PGM, হ্যাঁ? Similarly, now, so this PGM will convert glucose 1-phosphate to glucose 6-phosphate. What's next? It can produce glucose by like this, fine. But this enzyme is absent in muscle, fine. So in muscle, this is the last step. In liver, this is the last step. Followed? This is over. But problem is, this phosphorylase enzyme cannot cleave any glucose molecule beyond the branch point. Any glucose molecule beyond the branch point. Suppose it is coming from here, cutting one by one. When it comes here, it gets stuck, branch point. So there comes a second enzyme called activity. One is okay. One is alpha 1,6 glucosidase activity, another is glucan 1,4 transferase activity. Fine. 1,4 means the growing. আচ্ছা এখানে আছে তো, এখানে দেখা যাবে। See this diagram, na? This is showing two branch ends of a glycogen chain. This is one end, this is another end. These blue colored dots can be cleaved individually as glucose 1-phosphate by phosphorylase enzyme. I repeat, the blue colored dots can be cleaved individually by phosphorylase enzyme in form of glucose 1-phosphate. Beyond this, when it reaches 4 residues from the branch point, this is branch point 1, 2, 3, 4. This is branch point 1, 2, 3, 4. 4 residues away from the branch point, phosphorylase will stumble, will stop. It cannot act further. Why? Because the active site of the phosphorylase is a very large space. It accommodates 5 glucose units inside it. The first one, which is located very deep into the structure of the phosphorylase enzyme, can be cut. But here only 4 can enter. Its mouth gets stuck at this branch point, active site. So the 5th position remains vacant, khali, so it cannot cleave. Fine. So phosphorylase will stop here, and from here debranching enzyme will take the charge. It will transfer these 3 pink dots on any surrounding chain, these pink dots, and the green dot will be released in the form of free glucose. Green dot will be released in the form of free glucose. So, one glucose will be released in the form of free glucose due to this activity, and rest of these 1,4,1,4 glucon transferase activity, this 1,4 will be cleaved and here 1,4 will be joined. So, this chain will look like this, correct? This chain will look like this. Now, the pink dots are much away from the branch point, and if you remove this green dot, then there is no branch point anymore. So, phosphorylase, the previous enzyme, can cleave each one like this, right? And it can continue up to here. Clear? So, two enzymes for catabolism. Number one, phosphorylase, which requires inorganic phosphate. The muscle form of phosphorylase requires calcium as cofactor. The muscle form of phosphorylase requires calcium as cofactor, unlike the liver form. Unlike the liver form. So, the muscle form of phosphorylase has got a specialized supersecondary structure called calcium calmodulin structure, which is absent in the liver isoform of phosphorylase. Okay? Other regulations might be same for both the isoforms, but except the role of calcium. The muscle phosphorylase is calcium dependent, co-dependent you can say. The work is more efficient in presence of calcium than in absence. So, this one green dot is removed in the form of free glucose, and this one can be cleaved up to here. Okay. Without this kind of diagram, you cannot explain. So, whatever book you follow, read those in this way. Already explained, glucose 1-phosphate to free glucose, glucose to glucose 6-phosphate by phosphoglucomutase. Now, this diagram shows the fate of glucose 6-phosphate. Fate of glucose 6-phosphate. Glucose 6 can come from many ways. It can be oxidized from, it can come from glucose by hexokinase or glucokinase. It can come from breakdown. It can go for synthesis of glycogen. All this has been shown. Let us come to the synthesis part. Let us come to the synthesis part. How do you make a building? Then you need something to join bricks. So, what are the bricks? Glucose units. Slightly different. Only glucose cannot work as brick. You have to convert it into this form. Amay dekhis na. UDP-Glucose. UDP-Glucose is a molecule which carries glucose unit. UDP, what is UDP? Uridine diphosphate. Uridine diphosphate has come from UTP, uridine triphosphate, which is required for RNA synthesis. Correct? And uridine triphosphate is converted to UDPG. Previously, we have seen reactions ATP plus glucose forming ADP plus glucose 6-phosphate, where phosphate was being transferred. This reaction is opposite. Here the nucleotide is being transferred. Phosphate is released. Correct? Phosphate is released. So UDP is joined with glucose at the one position. It forms UDP-Glucose. So one of the function of nucleotide is as carrier molecule. What molecules it can carry? It can carry, you see, glucose in the synthesis of glycogen. It can carry gluconic acid in the synthesis of mucopolysaccharide. It can carry gluconic acid for detoxification of bilirubin. It can carry choline for synthesis of phospholipid. It can carry ethylamine as well, right? CDP choline. Fine. So, it is one of the function of nucleotide as a carrier for different molecules. UDP-Glucose is nothing but that. So, UDP-Glucose is the building block for glycogen synthesis. This is okay. Name of the enzyme? Pyrophosphorylase. Means what? It cleaves pyrophosphate. PPI means pyrophosphate. So, it cleaves pyrophosphate. These compound UDP-Glucose contain two phosphate groups. One of the phosphate was initially there, glucose 1-phosphate. Another came from UDP. Right? So, it becomes UDP-Glucose. UDP-Glucose. So that you don't misunderstand from the UDP sound that both the phosphates were contributed by UDP. That's why the name of the enzyme is so important. It is not phosphorylase, pyrophosphorylase. Clear? Fine. Now, after this building block is formed, the synthesis takes place in two different ways or begins in two different ways. Correct? Suppose you want to make a hut, small house, and you want to make a big house. If you want to make small house, then do you need to make a very big basement, deep basement? No. But you want a very big house, then making base is very important. It should be very deep, at least 1/3 or 1/5 of the total height. Right? So, in that case, the liver glycogen molecule is much bigger than compared to muscle glycogen. So, which one will require prepared base? Liver glycogen. What is the name of the base? Base means BAAC, means the primary RCC work done. That base, not acid base. Yeah? What is that? For liver, glycogenin. It is a base, protein based. Protein is a 50 kilodalton protein on which glucose molecules are added. Hello? But since muscle is not required. So, see, muscle glycogen has got third defined point. Third defines. First two I told. Second one, this synthesis does not require glycogen for muscle glycogen. Whereas liver glycogen requires glycogenin. It's a 50 kilodalton protein which is active when it is in dimeric form. Now, this one contains one tyrosine residues on which 3 to 4 glucose residues are added non-enzymatically prior to beginning of the glycogen synthesis. much before the glycogen synthesis takes place, the making of glycogenin or a compound on which glycogen can be formed called glycogen primer is formed. The glycogen primer is formed by glycogenin and 3 to 4 glucose units added to it, but non-enzymatically. No such evidence of physical requirement of enzyme has been found to synthesize glycogen primer till date. So, they called it non-enzymatically synthesized. Clear? Now, these UDP-glucose will go on joining one after another glucose units onto this glycogen primer. Fine? Very easy. Glycogen synthase will now come. What it will do? See, this is a glycogen primer, UDP-glucose coming, and it is attaching this glucose unit, red color glucose unit, and UDP will go out as it is. It will again get converted to UTP. That again UTP will take to UDP-glucose. Now, this will continue as long as it forms a very long chain. How do you count? At least 11 residues must be there. At least 11 residues must be there. Just count how many sugar units are there in this chain. It's around 15. See? Go. Huh? 15? 16? See? At least this is 1, 2, 3, 4, 5. 11 plus 5, 16. Now, these long chain, if glycogen exists as a long chain, it will look like amylose. We need to introduce branching. So, to break down branch, we have Debranching enzyme. To synthesize branch, we have Debranching enzyme. To synthesize branch, we have branching enzyme. Is it very difficult? So, branching enzyme, what it will do? It will take 1, 2, 3, 4, 5, 6, at least 6 residues from one end. Which end? Non-reducing end. This is reducing end. This is non-reducing end. Why they introduce this chain here? Just to make you understand which side is a non-reducing end. This is non-reducing end. This is reducing end. Correct? So, from non-reducing end, it will take up 6 residues of glucose units and transfer it to the neighboring chain and join it by alpha 1,6, at least 4 residues away from the end. At least. Very good. I am very happy. You understood everything. Now, this molecule has got 1, 2, 3. So, glycogen synthesis can continue synthesis further at these 3 branches. Again, when it will become very long, it will form branching, long, branching, long, branching, it will go on. And at the moment it will pass 2 hours from the last meal, it will get again shortened. Whole day it will grow like this. It will grow on, grow, again come back. It will go, again come down. Chain of the glycogen, whole day. Because every 4 to 5 hours we take something to eat. If we don't do, then this thing will not continue. Regulation of glycogen metabolism, very, very difficult and very, very important. We have regulation by hormones mostly. There are 2 hormones we will discuss. Obviously, one insulin, another. Fine. So, main enzyme for glycogen synthesis regulation is glycogen synthase. Main enzyme for, see, this is actually mistake. The name of the enzyme is not glycogen phosphorylase, it is phosphorylase. Those who are thinking this is the last slide, they are wrong. This is just beginning. After this slide, when I will actually go into the regulation part, I would request all of you to just concentrate very carefully. If you lose the pace, if you cannot go along with me to understand step by step, it will be very difficult for you to understand later. Okay, just for 10 minutes you be with me. Just tell me when you have finished writing this slide. Okay, there are nothing but informations. ATP, AMP, ADP, glucose 6-phosphate, what is their function, what is their role, that's all. Fine? We'll go. Just see this line, glucagon and epinephrine. Epinephrine, what is the function of epinephrine? Fight and flight response, right? So, if you want to fly, run away, you need more glucose or less glucose? So, glycogen should break down or synthesize? Break down. So, glucagon is just like epinephrine in this case. In this case, it will activate glycogen breakdown by activating phosphorylase. It will also inhibit glycogen synthase. Now, these 2 pathways, synthesis and breakdown, work as like a switch, electric switch. When switch is on, light is on, savings is gone. Switch it off, light is off, savings on. So, when synthesis continues, breakdown stops. When breakdown continues, synthesis stops. Insulin and glucagon and epinephrine, mostly glucagon, we confine to glucagon only. Glucagon regulates most of the enzyme by adding phosphate group, means phosphorylation. This is called covalent modification. Regulation of enzyme by covalent modification, adding phosphorylation. So, glucagon causes phosphorylation, insulin causes, what would Pakistan do? Opposite, dephosphorylation. Glucagon causes increase in blood glucose level, insulin causes, glucagon increases phosphorylase activity, insulin increases synthase activity. Clear? So, insulin causes generalized dephosphorylation, not in all cases, generalized, most cases. Glucagon causes generalized phosphorylation. So, wherever it will be regulated by glucagon, remember some protein must be phosphorylated. Wherever it will be regulated by insulin, remember some protein will be dephosphorylated. Clear? Keep this in mind for the next slide. This is called cascade pathway of hormonal regulation. Cascade pathway of hormonal regulation. What is hormone? Suppose outside Mr. Molloy is standing. correct? If I can see, then the hormone is something which is giving signal from outside. So she will go and bring the signal. So she will become a receptor. This is a membrane, there is hormone, and monocyte will say, okay, Sandeep sir, you have get out. You don't have to take class. Accordingly, I have to work. Huh? Fine. Similarly, when hormone comes at the doorstep of the cell, what is the door? The receptor. The receptor molecule binds to it, and the binding brings about some structural conformational change in the receptor, which is transmitted through the membrane to the other side of the membrane inside the cell. Inside the cell, there are certain molecules which are associated with the intracellular domain of the receptor. The receptor has got three domains: extracellular, intracellular, transmembrane. So, extracellular activity, if you do like this, what will happen? The tears will come down. Is there any connection? No. Connection is there. Twist, eyes become watery. See? Similarly, when you twist the outside extracellular domain, intercellular domain also will change, and that change will bring about certain changes inside the cell, and that will trigger a signal pathway or signal cascade. Cascade means step by step, like a water flowing from up to down. Right? So similarly, these reactions will take place starting from hormone attaching to the extracellular domain, then activation of something which is associated with the intercellular domain. What is that? G protein. Correct? The G protein when activated, G protein, when G protein is activated, it converts adenylate cyclase to inactive to active form. What is the function of adenylate cyclase? It converts ATP to cyclic AMP. Fine. What is the fate of cyclic AMP? Cyclic AMP is a midpoint of continuous synthesis and breakdown. The enzyme which synthesizes cyclic AMP is triggered by glucagon. An enzyme which cleaves cyclic AMP to AMP is phosphodiesterase, and this phosphodiesterase enzyme is triggered by insulin. So, when ATP is forming cyclic AMP, at the same point cyclic AMP is being broken down by phosphodiesterase. But in presence of more glucagon, which one will be more? Synthesis or breakdown? See, suppose outside in this door there are 10 glucagon molecule, and in this door there are 1 insulin molecule. So what will happen? 10 adenylate cyclase molecule will be activated, 10 cyclic AMP will be generated, whereas only 1 cyclic AMP will be destroyed. So 9 cyclic AMP will be accumulating. So this is a dynamic concentration difference of cyclic AMP, which will be sensed by the further down below the cascade pathway components. What is that? A protein called CDPK, cyclic AMP dependent protein kinase, AMP dependent protein kinase, CDPK, which is activated via cyclic AMP. When this protein kinase is activated, why there is no name, specific name? Because this enzyme can work on many targets, and there may be many such enzymes depending on the lower end of the reaction. What it is actually acting? Is it acting through this glycogen pathway, or is it acting some other pathway, lipid biosynthesis? So it will be different. Here CDPK will activate protein kinase. Protein kinase will activate phosphorylase kinase by all steps C, by phosphorylating. I said glucagon causes phosphorylation. So by phosphorylating it will activate enzyme. Which enzyme? The glucagon must increase blood glucose level. Which reaction? Breakdown. So the enzymes which prefer breakdown of glycogen will be activated by glucagon by phosphorylation. Is it clear? Now you need not to mug up which step is where it is phosphorylated, where it is not. You must remember the goal, ultimate goal of the pathway. What is the goal of the pathway? First two things you must remember. Glucagon causes phosphorylation, generalized phosphorylation. Insulin causes dephosphorylation, generalized dephosphorylation. Why I am using generalized? Because all the steps are not equal. And we are not discussing all the steps. Most of the steps are like this. Glucagon increases blood glucose level. So glucagon prefers glycogen breakdown. So glucagon prefers activation of phosphorylase. If glucagon causes phosphorylation and if glucagon causes activation of phosphorylase, that means phosphorylase is activated by phosphorylation. P, phosphorylase, P for P. Phosphorylase is activated by phosphorylation. What is next? When phosphorylase is activated, a counter enzyme is also present called phosphatase. But phosphorylase is activated not directly. It has a precursor molecule. What is that? Phosphorylase kinase. First, phosphorylase kinase gets activated by CDPK. Then phosphorylase kinase, active phosphorylase kinase will activate phosphorylase. Both the phosphorylase kinase and phosphorylase enzyme can be brought back to the inactive form by phosphatase enzyme. Phosphatase, water, entry of water, exit of phosphate, same reaction. So this is one cascade pathway. Clear? Regulation of glycogen breakdown or glycogenolysis. Clear? Let's write this pathway. Some different book might have given differently. I have shown another pathway also here. Much more easier. You want to remember a little less. Same thing. What about glycogen synthase? Glycogen synthase, remember I said it's like a switch. So when glycogen breakdown is predominant, there is no question of glycogen being synthesized. যখন ব্রেকডাউন হবে তখন সিনথেসিস হবে না। তাহলে গ্লুকাগন যদি ব্রেকডাউনকে প্রমোট করে, গ্লুকাগন কাকে ডিমোট করবে? সিনথেসিসকে। গ্লুকাগন যদি ফসফোরাইলেশন করে এবং ফসফোরাইলেশন করে অ্যাক্টিভেট করে, তাহলে গ্লুকাগন ফসফোরাইলেশন করে ডিমোট করবে। তাহলে কাউকে ফসফোরাইলেট করলে সে অ্যাক্টিভেট হবে। যেমন তুমি ঘুমিয়ে আছো, তোমার কান টেনে দিলাম, তুমি ঘুম থেকে উঠে পড়তে বসলে, ঠিক আছে? এবার তুমি কারোর সঙ্গে বসে মনের কথা বলছো, মান কি বাত, এমন সময় পিছন থেকে এসে আমি কান মুড়ে দিলাম। বা তুমি 10টা বন্ধুর সঙ্গে বসে আছো খুব অ্যাটিটিউড নিয়ে, এমন সময় মা বা বাবা এসে কানটা মুড়ে দিল। অন্য সময় মুড়লে অত ব্যথা লাগতো না, বন্ধুদের সঙ্গে মুড়লে অপমান বোধ, উল্টো হয়ে গেল। তাহলে আগে কি ছিল? তুমি অ্যাক্টিভ হয়ে গেছিলে, এখন কি হলো? তুমি রিঅ্যাক্টিভ হয়ে গেলে। হ্যাঁ, কোনো সেন্স নেই, কার সঙ্গে কি করছো, বড় হয়ে গেছি আমি এখন, তাই না? হয় কি না? So same phosphorylation can promote some enzymes, same phosphorylation can inhibit some other enzyme. The enzymes which are involved in synthesis of glycogen will be inhibited by the same action, phosphorylation. Correct? And here insulin will come as a savior, because when glucose is plenty in the blood, then only glycogen is synthesized. When glucose is plenty, insulin secretion increases. So outside here, now there will be 10 insulin molecule and 1 glucagon. it may be here 10 glucagon molecule is there, but here 100 insulin molecules have come. The ratio is same. The cell will see 10:1, be it 1000:100, be it 100:10, be it 10:1. Ratio is fine. So, in apparent increase in insulin concentration outside the cell, remember the receptor mechanism of glucagon and insulin are different. That's why I mentioned two different doors. Correct? Not in the same door. Their doors are different. That is why their actual ratio is not there. Their কি বলে এটাকে, মানে অ্যাকচুয়াল রেশিওটা দেখা, মানে কি বলবো ওটাকে তোমার, হ্যাঁ অ্যাপারেন্ট রেশিওটাকে দেখা হয় আর কি, ঠিক আছে, অ্যাকচুয়াল রেশিও দেখা হয় না, বা কি উল্টোটা বললাম আর কি, মানে এম্পিরিক্যাল ব্যাপারটাকে দেখা হয় না। যাও গুলি গেছে ওয়ার্ডটি, যাও ওটা বুঝতে পেরেছি আশা করি, হোপ ইউ হ্যাভ আন্ডারস্টুড। নাও when this occurs, glucagon is absent. Rather, we show like this how glucagon inhibit glycogen synthesis. Can we show that? Because insulin pathway is little bit difficult, glucagon pathway is much more easier. Fine. So glucagon attaches with the receptor, it brings about changes ATP to cyclic AMP. Cyclic AMP stimulates what? CDPK. CDPK phosphorylate glycogen synthase. But in this case, phosphorylation makes it inactive. In phosphorylase, phosphorylation makes it active. In synthase, phosphorylation makes it inactive. So glycogen synthesis will stop. The other enzyme which brings back the glycogen synthesis to active form is protein phosphatase. Protein phosphatase, which is so when insulin molecule is more in number, this pathway will be predominant. More and more glycogen synthase will get dephosphorylated. More and more glycogen synthase will get dephosphorylated because insulin is more, and it will synthesize. Long question, one question is left, the last slide. The last slide will take, one class is required to take. Tomorrow most probably HOD sir will take if I am unavailable. Okay. Any questions so far? From here only one question is important, both the pathways and both the regulation.